Welcome to a special episode of the BioCentury This Week podcast. My name is Josh Berlin, and I lead corporate alliances and business development for BioCentury. We are taping this episode live on stage in Seattle, where we are wrapping up BioCentury's third Grand Rounds-U.S. conference. For those of you who don't know, Grand Rounds is BioCentury's early-stage R&D conference, where we focus on how to overcome key bottlenecks in translation between academia and industry, and how to make early-stage R&D investable. We've had 2+ days this week of debate and discussion here in Seattle, plus a track of early-stage presenting companies and an academic poster session. And there's just been a lot of energy this week from my perspective. So first off, on behalf of BioCentury, I just wanted to thank everybody here in Seattle who's been so welcoming and so generous with their time. I want to especially thank our regional hosts, the Allen Institute, Fred Hutch, the University of Washington, and Life Sciences Washington. We would not have been able to do this event without their support and everybody else in the community. I also wanted to give a quick shout out to some of our previous regional hosts for this event. Previously, we were in Chicago. Before that, we were in Nashville. We just announced today that next year we'll be in Houston. And I thought it was just really great to see folks here in Seattle, talking to folks in Houston, talking to folks in Nashville, talking to folks in Chicago. And it's really, I think, Simone, this is really what we're trying to help do is sort of foster interactions between ecosystems and so, allow people to build connections.
So Josh, you're kind of, for me, like the Pied Piper. You know, you do this and then you pick them up along the way and they're all fun. But seriously, we're really building a community from these different Grand Rounds events. But the other thing that I think we're going to tap into as well, it is our third US one. We, of course, have had one in the UK and have one in Amsterdam, so we're doing a Europe version as well. But it is very interesting to see some themes, how some evolve, even in this short time. Guess which ones? Super fast, and how some seem to take a little longer to resolve. But yeah, and you know, I know you're going to sort of... sorry, I've got Aaron to my right here from the Allen Institute.
Let me introduce the team here. So we obviously have Simone and Selena, who everyone on the podcast knows. And we're really happy to have Aaron Coe, Managing Director of the Innovation Portfolio at the Allen Institute, who also served on the Regional Host Committee. And we have Sam Blackman, Entrepreneur in Residence now at GV. I'm sure everybody here knows Sam from day one Biopharma. Also has been at several multinational pharmas, as well as a few biotechs here in Seattle, including Seattle Genetics and Juno. So really welcome them, and we're looking forward to sort of getting their take. Aaron, maybe we'll start with you as the regional host spokesman. I mean, you know, a lot of energy this week. Again, couldn't have done it without you and Arden and the Allen Institute, as well as our friends at the Hutch and at Life Sciences Washington, UW. Tell us from a Seattle leader's perspective, you know, what did you see this week? What did you hear? What's your takeaway?
Well, longtime listener, first-time caller. This has been a wonderful event. The Allen Institute is extremely happy to have sponsored this, and we are very grateful for BioCentury for building this network of networks. I think we are all speaking one language, which is scientific productivity, and we speak with many different dialects, whether it's from where we come in the country or around the globe, but also from where we come in terms of the stages in which we're contributing. And so to bring the institutes of Seattle together to be able to kind of really, really shine a light on all of the interesting innovation that's happening here is really a wonderful thing. And to bring people together with folks further down the value chain has also been a really powerful set of conversations, and we couldn't be happier with the energy that we were able to see and all of the really fun events that you guys put on. Last night we were at the Chihuly Gardens Glass Museum, and
socks.
It was insane. Oh, those come from the nice. Yeah.
Simone, what kind of socks are those?
They're Chihuly socks.
Oh, those are the Chihuly socks. Okay, so
That's
did
a good
walk
purchase.
us through the gift shop, I believe, as we were going in. So they got you. Yeah.
That's good. Yeah, no, being able to break down kind of barriers between folks and have them meet in the halls, meet in front of posters. We had a great group of our rising star scientists from every single one of our accelerators be able to be represented here and interact with folks all across the industry. So it's really 115 organizations that you brought into town. So this is really something that stimulates a lot of great interactions and partnership opportunities. we were really, it was right on the heels of a huge announcement for our organization. I'm just incredibly grateful for all of the attention that's being paid to some of the great work that's being done by my colleagues. So
thank you. So, the first night of this event, we had it at your institute. It was a beautiful building, and we kicked it off, with, I thought, a great keynote and discussion with David Baker. I mean, Simone, what was your take on David Baker's talk?
So actually, can I just take a step back? Because, Selena and I talked about this. So we had David Baker, who's the 2024 Nobel Prize winner. We also had Mary Brunko speak as the 2025 Nobel Prize winner, both of them from Seattle, right? Or from the local area. So two more different people and journeys I can't imagine, right? And so, this was really interesting for me because I think it also captures how science works. So on the one hand, you've got something very directed. I mean, David Baker, and we'll talk about it, has just some phenomenal things with protein engineering and the use of AI and so on. And Mary Brunko gave exactly a different story about something that, discovery in 2001, that now that, let's say FOXP3 and Tregs, that now we're beginning to understand the translational value. And that is science. On the one hand, it's directional and you can move it to accelerate things. And on the other hand, sometimes it just takes its way. And I just thought that was a very kind of interesting juxtaposition.
Yeah, I think one thing that was remarkable about his talk is the number of projects that have spin out potential applications in the real world, and they're really diverse, and there's a large number of them. So just that one lab alone tells you there's a lot of substrate in Seattle for biotech, even if there are hurdles, right? And I just want to give a little plug to also Jay Shendere's talk. This is another one that's super visionary, and you can imagine further in the future, these DNA typewriters, where you can encode, theoretically, hundreds, maybe even thousands of cellular events simultaneously over long periods of time, putting together that entire cellular context in which important events are happening has also many potential applications.
Sam, thanks, Selena. Sam, I wanted to ask you something. So we've been doing this event now for several years, and something our CEO, David Flores, pointed out at lunch today. It's I almost think every panel this week, AI was a key focus, regardless of whether that was what was said on the title of the session. Much different, I'd say, even than a few years ago when we kicked this event off in Nashville. So what's your take on AI and what you heard this week on the various panels about how folks are thinking about AI for drug discovery and development?
Thanks. Well, first, thanks for having me here. It's nice, even though I no longer live in Seattle, I live within the penumbra of Seattle, so I'm always happy to be able to come to a meeting that requires something less than a transatlantic airplane ride. My recent transition from being an operating role over to Google Ventures has been nothing short of a 10x or 100x increase in the amount of information that I'm now aware of regarding artificial intelligence, and that's both on the pure tech side as well as on how it intersects with either drug development, drug discovery, or payer-provider work. It's pervasive throughout the entire ecosystem now, and whereas 5 or 6 years ago, or even 3 or 4 years ago, when we would see vendors and they would come and they'd pitch us on their various services, and I'd see pitch decks for things that I was asked to advise on, and there was the sporadic sort of, we're going to do this with AI/ML, and you would ask people what that meant, and they would say, well, we have some algorithm that we're using, and you'd pull on that thread and say, tell me more, and then you'd ultimately end up in some very hand-wavy, we think that there's some machine learning way for us to do this aspect of drug development faster. In a very short period of time, and I was listening to the Hard Fork podcast. I'm sorry to betray you for another podcast, but I was listening to Hard Fork, which is my favorite tech podcast, and it reminded me that ChatGPT showed up in 2022, so it's only been 4 years, and what we've seen now in the past 4 years is essentially an end-to-end retooling of how we think about drug development and the inclusion of fully mature or increasingly mature artificial intelligence systems in every aspect of what we do.
Sam, I want to pick up on that because one of the threads that came up, so there are some things that change and some things that seem not to change, right? Certainly the pace at which of AI fluency, like we all know what machine learning means now and so on, right? But one of the things that came up in many of the sessions was the need for more data, was the need for negative data, was the need for more rigorous data and data sets. And I have a few of my own thoughts, but we've been having this conversation for so long, we meaning the ecosystem, has been having this conversation for so long. And so I guess my question is whether they're, sitting in GV, is there really a sense that what's changed now is actually our ability to harness that data if we could sort it out, right? If we could get the data in there, and we've talked about making it interoperable or federated systems or whatever, but is there a different belief now that was coming through to you in what we could do with the data because of AI?
Oh, absolutely. I mean, I've always been a big sort of large data set, empiric, top-down. There are patterns and we just can't see them. And this may betray my roots as a scientist. I do actually, did have some scientific training at some point in my life, but I'm something of a neo-empiricist. I believe that you can understand the world equally well with a top-down approach as you can with a bottom-up approach, and that's a completely different philosophical discussion. I think that we're fully bought in on that we generate so much data now of such depth and complexity that there are patterns in there that we can't see, that the machines can tease out for us that are going to enable new ways of seeing or thinking or doing. Within that, though, there's a fundamental problem, right, which is that we're only as good as the data that we have available to us to train these models on, and that data, to some extent, comes from things that we produce and publish and codify and put into data sets. And we're biased in how we do that. And so I think somewhere along the line, this conversation around the reliability of the output is going to be proportional to the quality of the input data that we have. And right now, that's
just
biased.
To name it, you're talking about the missing negative data mostly.
Absolutely.
But I want to pick up on that, again, drawing on something that came up in one of the panels. So the missing data are the data that aren't there. But the other thing that kept coming up is, garbage in, garbage out, the quality of the data. And I kind of feel like AI needs to grow up and be able to sort out good data from bad data. And that was sort of this idea. And you're talking about patterns. And so I sort of wonder whether there's this sense that any of you got that, just put it all in there and let the AI learn rather than how much curation is going into. I don't know. Selena,
I think that comment was made on the Transformer panel by one of our colleague Lindsay's panelists, that actually once you get all that data in there, it's so good at finding patterns, it can actually find the mistakes in there, the anomalies, the bad data, and pull that out as separate. So yeah, I think so.
That was a great panel, actually, too.
Yeah, and Aaron, I know you've also been making the point that some of the institutes have a role to play here in terms of data, right?
Yeah, we have hundreds of petabytes of data, and we spend an incredible amount of time and effort in the quality assurance of that data. So at the earlier stages, I know there's some problems at later stages with what's missing from the clinical set, but one of the things that I think our announcement that came on the 2nd with the announcement of the Brain Health Accelerator is this notion that we have a tech stack that is very robust, but we also have achieved this level of leadership in the community to be able to bring consortia together, to bring data together. And sometimes these different areas have siloed data. And so what we're really excited about is to be able to kind of be a marshal for some of these efforts that will get, expansive in terms of looking at neurodegenerative diseases.
And it sounds like, though, there's still some work to be done on creating, I think, what somebody called tiger teams, like people who are biology natives or tech natives, because it's interesting. With select people here, you can hear the conversation turning toward, okay, well, how do I create that harness around the model for my thing? That's a term you'll hear in tech much more so than like our industry, or like, how do I do a very good job of creating that orchestrated set of agents with the good orchestrator at top? And a lot of people don't know how to do that yet. So there's different levels.
But Selena, going back to the point earlier, I wonder if in Grand Rounds in 2 years' time, we're all going to be fluent in that too.
I think so.
Okay, so we talked about the first day, AI. Another thing I think we heard a lot about this week is another A, animals. And so we heard a lot about preclinical models, a lot of debate, a lot of vigorous debate on stage about where the future of animal models are going, where NAMs are going. I know, Selena, you and Simone spent some time on stage with Matt Hewitt of Charles River. I thought it was a really good discussion. So what's your take? What do you hear this week that maybe surprised you about where the future of preclinical models is going?
Surprised me. I don't know about that, but I definitely heard a lot of cognitive dissonance, a lot of the animal models are crap. Actually, the animal models are really predictive, or we need to move away from animals. No, we need more animal models. I think people are just kind of all over. And it depends, of course, on the application, because what you're using, there's a lot of different things you use the models for. But I don't know. Do you want to?
Actually, I want to throw it to Sam. I mean, yeah.
Well, no, I will tell you.
Where are you? Animal models, good, bad, terrible, more, fewer, NAMs. What are you?
I mean, what's the old aphorisms, right? All models are terrible, but some models are useful. I mean, we could spend the next hour reinforcing the long-held, I think, and correctly held belief that one of the biggest barriers to drug development that we face is the lack of predictive translatability of our preclinical models, right? I think the negative predictive value of many preclinical models is high. I think the positive predictive value is low, and it's evidenced by the fact that our success rate going from preclinical to proof of concept is not 100%. And we have limited numbers of resources to be able to, invest in building more and more reliable models. We continue to do better with organoids and slice models and things that are close to human tumors, but they're all going to be pretty terrible. And at the end of the day, it's going to be very expensive human beings that are going to tell us whether or not a new molecule or a new mechanism is working. That being said, one of the things that I loved out of the Charles Rivers presentation was the fact that they're using natural history data to reduce the number of animal models. And boy, did that make me happy. And it's such a no-brainer. And as soon as I heard it, I was like, how come we didn't think about this? How come we didn't think to just gather data from thousands and thousands of dogs and just have a synthetic control arm of dogs and cut down on the number of animals that were just placebo dosing? It was wherever the Charles Rivers guy is. If you're
Matt,
out there,
where are you?
well done, you. I mean, well done, Charles Rivers.
Totally, that would go under the surprise thing. And we Selena and I had seen the slides beforehand, and it's effectively digital twins. It's animal digital twins. And yeah, you're absolutely right. But you still come back to that, maybe for tox or whatever, they're still like crap models are crap models, whether they're animals or not. But it is a very short jump from there to first in human, right? Because it's all about what you just said.
Yeah.
It's like what you need is something that's going to be predictive.
Right.
Yeah.
Okay.
So that was my third A. That we had AI, we had animals, and the third A was Australia. Which
Oh, I see.
I think has never been said more on stage in biotech, and that's sort of a shorthand for first in human studies. And Simone, I thought you had just an all-star panel here on stage in Seattle. We had the head of the Fred Hutch, we had the head of Texas Medical Center, we had the head of the Parker Institute, former FDA leader, and you sort of leading the charge. So tell us a little bit about that session. And there were so many takeaways, but just pick out one or two.
So let me just say, first of all, we are going to write about that. So we will talk about some of those takeaways. It's interesting. There were a lot of passionate voices on the stage and then off the stage afterwards among different people on this topic. And so various things, if I think about it, so we had the heads of two major medical centers, right? Texas Medical Center and Fred Hutch, okay? And we also had a regulator talking about what regulators can do. And we had Karen Knudsen, who represents the Parker Institute, but really was channeling the patient voice actually in this. And so there are various things all the way along the line that are obstacles and hurdles for why it takes so long. When we talk about first in human, it is partly getting the IND approved, but it's really to get the human data. That is what it is. It's like from from application to human data, right? And a lot of the things on the way seem to be obstacles or hurdles inside academic medical centers. And so some of it relates to protocol amendments and how long that takes. And in some parts, there's a regulator role, but some of these things are really just up to the centers themselves. And so that seems to be the good thing that's come out of this, or one good thing is we start to see an alliance between them, a meeting of minds from different medical centers that actually might be able to do some sort of, learning or best practices or something. And then there's a whole different thread, which is what does the ecosystem and the regulatory system need to do to make, I'm gonna go there like back to Australia kind of thing, make the US competitive, to make innovators want to run their study here or stop fleeing to do their study elsewhere. And I know you've got different thoughts on this that you might want to air some of them,
Sam. I think you have some thoughts about this. Please, keep it PG-13.
They're a little wishy-washy, though. They're not.
I have thoughts. So, look, I've been doing phase one for
I have thoughts, he says.
Thoughts. and opinions. I've been doing phase 1 research for almost 20 years, and nothing that came up in that panel discussion was news to me, and they're not new problems, and nor is the idea of going to Australia for some time savings. And by the way, it's not years of time savings, it's months of time savings. I actually think that the US IND system, if you work through it properly, if you have a pre-IND meeting or you're going in with a drug where the mechanism of action has been well characterized, and you write a clean and responsible and thoughtful protocol, and you interact with the FDA appropriately, you can get your IND cleared in 30 days. I mean, it happens automatically unless you've done something wrong. And I think if you do things right, then the system works. What gets annoying and expensive, both in terms of dollars and time, is the nuts and bolts stuff. It's all of the site activation steps required at a site, and each of those is expensive and requires human interactions. It's the budgeting and contracting for which there are no universal templates. And every time you're going through this exercise, if you're running a trial at 10 different sites, you're having 10 different budget and contract negotiations with 10 different formats. You're having site qualification visits and having to inspect every single pharmacy and where they store the investigational product, and you have to train the staff and so on and so forth. And you've got to run the gamut of 10 different scientific review committees and 10 different IRBs for the same protocol. And these are all, for the most part, NCI Conference of Cancer Centers. They're all, experts in what they do. And at the end of the day, and I think Tom Lynch brought this up in his talk, if you get through the IRB at MD Anderson, from his perspective, that's good enough for him. It should be good enough for the Fred Hutch. I thought that was a brilliant insight into a way in which we can easily cut out months of time in not the IND part. The IND part's short. It's the study startup part that is killing us.
Yeah, I thought that point on stage, and I think Karen of the Parker Institute also mentioned it, is sort of using NCI designation perhaps not only as a carrot, but also a stick, in the sense of if you're in that class, you should be able to recognize.
Well, yes, and because it does require resources, and it's to some extent an unfunded mandate. It's one thing if we say to be an NCI Conference of Cancer Center, you've got to start a protocol within 6 weeks of getting the regulatory document packet from the sponsor. Sure, it's doable, but you need to resource all of the systems there to be able to do that. And of course, we're doing the opposite of that today. We're de-resourcing our NCI.
Right, and so the overhang of this is obviously the way that China has just bent the curve on this and changed the pace. And so I think that you're right that a lot of these issues that were discussed on the panel are not new issues, but there does seem to be, it's sort of a little bit like AI's just created a new motivation to, let's say, get the data or whatever. This has created a new urgency to solve this, because I think people are looking at that efflux of potential going elsewhere.
Well, I think what we're realizing is that there are faster and cheaper ways of doing this. Now, they're not perfect. And of course, Endpoints and Statin, every other newsletter this week was talking about, now the federal government is saying, well, wait a second here. We're watching China, generate all the molecules, and we're watching companies running to China to run early phase clinical trials. And they're about to do what government typically does, which is have some type of blunt tool to fix what is a very subtle set of problems.
I would encourage you to read what BioCentury is writing about this, of course. Right?
I wasn't reading any of it. I was just, I was paying rapt attention to the panel so I could be prepared.
Claude was reading it for you.
My agent was reading it for me 100%. But I think the thing about China is you've got very large institutions that are running investigator-sponsored trials where the data is not necessarily going to be fit for submission to the FDA and needs to be verified. And there are problems with there's certainly advantages time-wise and scale-wise to going to China that we can't replicate here in the United States with the current system that we have.
Yeah, and so, Josh, I'm actually very surprised you haven't plugged our China conference at this point.
Yeah, please.
Which takes place 2nd to 4th of November in Shanghai. But I think, that's a conversation that I anticipate, that and the animal models one. This is a conversation that I anticipate being in future Grand Rounds for a while, right? But I thought, there were so many really deep science conversations. I thought your precision neurology panel, Selena, had some interesting things. Do you have any top take-homes from that one? You're going to kill me for that question? Okay.
That was one of my favorite panels, by the way.
Do you have take-homes from that one? Because you were in the audience.
Okay, everybody, this is how it goes, right?
Well, yes.
So, Aaron, did you have any take-homes from that panel that Selena ran?
Well, I thought it was extremely well run, and I was very proud to see my colleagues on the panel. The thing that struck me was the positive tone in terms of how each of these different contributors had aggregated tools that are now kind of coming into their own. And so I encourage everyone to sort of follow what we're doing at the Brain Health Accelerator, because there's a lot that is going to be moving in a very translational direction relative to our history and building all these, foundational data sets. And so it's an exciting time.
Yeah, I think it's clear from that. I think somebody used the word humility. You do have to have a lot of humility, because incidents like capsidas, we can't predict just yet. They might look good in a non-human primate and then, kill somebody in the clinic. But when it comes to avoiding places in the brain where you don't want your transgene because it's going to cause a problem, or needing to have a low dose that you can actually manufacture, there are several layers of precision and a toolkit across each one that you can kind of mix and match and tune to, even with the limits that we have today, start getting into precise brain regions. And I do think that's a very optimistic message.
Yeah.
Can I just call out another couple of panels? So first of all, I want to thank a couple of our other moderators, Melissa Bonner for a really interesting CAR T panel, and Rob Hirschberg for a really interesting panel on, I guess, what makes it investable, what makes early science investable.
Can I just say what his solution to all of you looking for early stage investment was, I believe, to start an AI company to de-risk assets out of China that have had fast proof of concept, and then you're good. You're good.
Problem solved. Yeah.
But also a couple of really key workshops, like a target product profile workshop, which I think is just like I don't know if I should say under discussed, but this is a thing that I really feel that the academic industry interface is a really important place for that conversation to start. Sam.
Yeah, I mean, I saw that a theme that we haven't touched on here that ran across the whole meeting, certainly anytime an investor was up here, was you need to tell me how this becomes a product. You need to tell me how it becomes a product, even if you're just talking about an idea. During the session on the DNA typewriters, I thought that there was a really interesting set of interactions between the scientist who was leading the technology and investor who was sitting there and trying to understand, I don't understand how
would you go about evaluating?
How is this investable, right? How does this generate a return on investment? And you literally it reminded me of something from when I was at Pharma. We used to joke, this was when I was at Merck, that the scientists would make something in the laboratory and sort of throw it over the transom, and they would think it was perfect. And we on the development side would catch it and be like, what am I supposed to do with this? And to some extent, it was like that with the scientist and the investor, which is, I've made this thing, and now I'm going to throw it over to you because I want to. And the investor's like, I don't know what to do with this.
There's a whole lot of bridging still needed there.
And so the TPP, the thinking about how does this turn into a medicine for somebody as early as possible is one of those themes that ran across all 3 days of the meeting.
Yeah, I mean, I do know an academic innovator who did start a company who would then tell the story that they did this incredible work and they published it in Nature, and then they sat by the phone waiting for it to ring, kind of thing, And so I think that probably that was a while.
Jay is not sitting by the phone waiting for it to ring. He's making a lot of phones ring for sure.
No, but I think you're right. I think that interface has really come a long way since the story I told. But I think there's still a lot to understand, because the concept of a target product profile is so often thought about something that is just industry relevant and, happens from phase 2 on, and it's all about commercialization. And so breaking down some of that is really interesting.
I think that's spot on what you're saying. There was a lot of folks in the room this week that want to be first-time founders, brilliant scientists, but haven't done this before. And so that is one of the missions that, Selena and Simone and the whole BioCentury team is really trying to help further along this discussion, trying to help get folks like you, Sam, like you, Aaron, and others in the room together to learn from each other and to talk about how to build companies and how to take medicines from discovery all the way to patients. Yeah.
And to that end, I think these last 3 days were a tremendous success. Lots of discussions in the hallways, just like you'd set this up. So
yeah, so
kudos.
It is what we're trying to do. I did have one investor tell me, they actually met a bunch of companies and they are going, company wannabes maybe, and they will have follow-up calls. We had how many posters? We had like, I don't know,
We had,
30 plus.
I think 35 posters. We
Right.
had 25 presenting companies, most of them pre-seed or seed companies.
Yeah.
Some with some experienced founders, some with first-time founders. And I
And
thought
a
it was,
rising star that we wrote about in BioCentury.
and this year's rising star was actually not, what was it last time? It was a setting sun in Chicago.
Yeah, but
'cause we had
I told Josh, so, the image that came with the story was of this beautiful statue, almost like an Oscar. So now he has to go out and source. We'll need a sponsor for the Rising Star statue.
That is true. So if anyone listening on the podcast wants to sponsor that, please send me a note on LinkedIn. We'd be happy to speak with you. So we're almost out of time. I did want to also talk a little bit, just real briefly, about where we're going next. So Simone, our next Grand Rounds event is our second Grand Rounds Europe. We're going to be in Amsterdam, September 23 to 25, with Forbion and BGV as our regional host sponsors and committee sponsors. So what are you looking forward to? I know you're going to have a discussion with Emer Cook of EMA.
Stole my line, Josh.
Yeah.
You stole my line,
Okay.
yeah. So first of all, who doesn't want to go to Amsterdam, and who doesn't want to go to Amsterdam in September? So I'm very excited about that. Yeah, so we have Emer Cook from EMA, who I will be having a conversation with. If you don't know her, she's a good conversation. She's an interesting and lively person to speak to. And we are putting together an agenda which we're actually going to post. We'll be posting that in this coming week. I won't reveal too much now about the nature of some of the other sessions. We hope to have a debate. Let's see. But we will have, as we traditionally do, some very kind of in-depth, it's a science conference where we're really talking about translational issues that are coming up, like bottlenecks. And so we're very excited to engage with Forbion and the local ecosystem. I think we're talking about the Benelux kind of area, right?
It'll be a showcase for the Benelux region. So Sam and Aaron, we expect you out there. We expect everyone here in the audience out there. We'd love to see you. And then we will also, as we announced today on stage, be back with this event. The U.S. edition of Grand Rounds will be in Houston next April, working with the Texas Medical Center. It'll be a showcase for the whole state of Texas, and we hope you'll all come out and join us for that. So I think with that, just wanted to thank Aaron and Sam, obviously Simone, Selena, and our whole team, which just did an amazing job this week. And thank you all for being here and for being part of the discussion and the dialogue and the debate. And we hope you'll join us again next time. So thank you.
Thank you too, Josh.